All right, are there any questions for the last week, from last week? I have your quizzes. Xavier gave them to me, and you guys did okay. You guys did pretty good. Not bad enough that you're going to need another normal value quiz. So I was pretty happy with how that went. So so we'll just move on to abnormal red blood cells. Okay, are there no questions at all? You can just jump right in if you like. Also, too, I sent your homework back. I sent your homework back. Most of you did pretty well. A couple of you confused leukemia with bacterial infection. Remember, leukemia is a really high WBC, and bacterial infections are in the 20s to about 50. Okay, and then your quiz tomorrow. It shouldn't be too bad. I'm going to be sending that to Xavier tomorrow. He'll give you the quizzes tomorrow on 3 and 4, and then I'll correct those. All right. So far, everybody's doing okay from what I see from the homework and from the quizzes. Okay, if no questions, abnormal red blood cells. As you know, variations in shape is poikilocytosis. So unlike anisocytosis, if you report poikilocytosis, you have to report the type of poik. So if you see a smear where there's a lot of different shapes, you can't just say 2 plus poikilocytosis or 1 plus poikilocytosis. You have to say that there is poikilocytosis, but then you'll have to enumerate what you're seeing. You need to say there's target cells, there's teardrop cells, there's schistocytes. Those are all the different types of poik or poikilocytosis that that you're seeing. So you just can't report out, well, there's 2 plus poik and then move on. You have to tell the doctor what type of poikilocytosis you're seeing. Okay, spherocytes. Spherocytes are shaped just exactly how it's the term states. These are spheres. These are balls, and the way they look like is that there is no central pallor, and their MCV is low. So these are small. So I'm going to be showing you a slide later on what spherocytes actually look like. Okay, smaller surface size. They're sphere in shape. Increased hemoglobin. And these cells are formed as a result of a defect or loss of cell membrane. And the one thing about spherocytes that you need to be aware of is that I know I've been telling you about high MCHCs. If the MCHC is greater than 36, then you need to suspect that there's some kind of artifact that needs to be corrected. In the case of spherocytes, these are true cells. These are not, it's not really an artifact, but it will be about 36, 37 MCHC, so you may be confused. These cells, this type of situation cannot be corrected. You can't, it's not an artifact. I mean, patients will have spherocytes in their blood, so it can't be corrected, cannot be, all right? Okay, so troubleshooting. I've dedicated a whole lecture 10 on troubleshooting, but this is kind of like an introduction. So I told you that the MCHC is one of the first things, if not the very first thing that you look at when you're reviewing an MCHC to determine acceptability. So if you see a high MCHC, then the steps in troubleshooting, the first one would be inadequate mixing. Hopefully, if the tube is not mixed adequately, that could result in a high MCHC value. So mix the tube and repeat the results. Another easy way to troubleshoot a high MCHC, it could be the caused by cold agglutinins. And the easy way to correct for high MCHC in the case of cold agglutinins is to put the tube in a heat block for about 30 minutes and then repeat the test. That's a common one. Patients will have cold agglutinins resulting in high MCHC. All you have to do is just put the tube in a heat block for half an hour and then repeat it, and then your MCHC should come down to normal. So that's a more common one. Another common ones are, well, less common would be lipemia, high protein, or autoimmune antibodies. In the case of lipemia, okay, so you have a purple top and it has a high MCHC. So you put it in the heat block and you repeat it on your analyzer and your MCHC doesn't come down. Okay, so your MCHC doesn't come down. So you now ruled out cold agglutinins. So what you have to do is take a sample of the blood and spin it down and try to find out what's going on. You need to take a look at the plasma. You spin it down and take a look at the plasma. If the specimen is lipemic, usually the MCHC is high because the H and H didn't match. So the second thing you look at with a high MCHC is you take a look at the rule of threes to see if it'll pass. Remember the rule of threes. Hemoglobin times 3 is hematocrit. Okay, so in the case of high protein, and I should also include icteric. Icteric is another one. Lipemia, icteric, or high protein autoimmune antibodies. If you spin it down and you see a white milky substance in the plasma, those are chylomicrons. That's lipemic. That is a lipemic specimen. Okay, the plasma is not, usually the color of plasma is a light yellow, and it's not amber. Okay, it's a light yellow, and it's clear. Okay, that's normal. If it's lipemic, that means the plasma looks like milk. It's really thick and it looks like milk. It's white and it's really thick. And that's why the hemoglobin reading is really high in comparison to the hematocrit. Your rule of threes will fail in the presence of lipemia. Okay, if your specimen is lipemic, you look at your MCHC, okay, something's going on, it's super sky high. Next thing you do is you look at your H and H. Your hemoglobin relative to your hematocrit, the rule of threes will fail. So the hemoglobin will be sky high because of the lipemia. The lipemia is caused by chylomicrons. When you get into chemistry and you go into the lipid chemistries, your blood has a high lipid content. I mean, if you eat, somebody eats like 10 Big Macs a day, your blood's going to be lipemic. Or you eat nothing but pork, your blood's going to be really, really lipemic. It's not good for your health. It's not healthy. So what will happen is you're going to have a high lipid content in your blood. That's lipemia. Another thing is called Icterus, and I touched upon that last week. If you have liver disease and you spin it down, okay, so you spin it down, it's white, it's lipemia. But if you spin down a sample of the blood and it's really dark amber green, orange, it could be orange or greenish, or it's usually greenish, or it's kind of dark. Okay, so remember, the color of normal plasma is yellow to light yellow. But if you spin it down and you see it's kind of like dark greenish, really, really orange green, that's Icterus. Okay, that's called Icterus. You guys know the spelling? You want me to spell out the Icterus? Let me see if I can spell that for you. Because these are terms that you probably have never heard of before. Let's see. Okay, Icterus or icteric. If you see that it's green or dark green or really dark amber or even reddish orange, I don't want to say reddish orange, but it's greenish, more on the greenish side, that means the patient has a liver problem. Okay, that's going to cause that's going to cause the hemoglobin to be giving you a high reading. Okay, it's going to cause the hemoglobin to be giving you a high reading. And autoimmune antibodies, that's not common. High protein. High protein is another one. That's really the third one. Do you remember a disease that results in a high protein content? Somebody name one. Anybody? Begins with an M.
Would that be multiple myeloma?
Yes, multiple myeloma. That's one. The other one is like Waldenström's. Waldenström's macroglobulinemia. Those are the two main ones that will give you a high protein content. Now, this is a tricky one because you're going to spin it down. You're going to spin it down, and it's going to be light yellow. Okay, but you have a high MCHC and you have a high, your rule of threes will fail. And this actually happened to me. So I spun it down. The plasma was light yellow. MCHC was unacceptable, and the rule of threes failed. So what I did was, I didn't know this, but I said, okay, well, I'm gonna go do the saline replacement. And that's the way you troubleshoot it. That's where you take an exact amount of plasma. You take, after you spin it down the sample, you take an exact amount of plasma and replace it with saline. Okay, that's called a saline replacement. In the case of Lypemia, again, you take an exact amount. I mean, it could be 500 or 700. If you take out 500 or 700 microliters, then you have to replace it with 500 or 700 microliters exact, okay? And the same thing for Icterus. You take out 500 or 700, it doesn't have to be 500, 700. It has to be whatever arbitrary amount, you replace it with that same amount. That's called a saline replacement. So you're taking out the junky part and replacing it with something that's clear. In the case of this high protein, it looked like I'm taking out good plasma and replacing it with saline. So what happened was, lo and behold, I reran the sample and the MCHC came down to normal and the rule of threes passed. The hemoglobin times 3 was hematocrit. So that's one where you won't, the problem is not visible, like high protein or Icterus. High protein or Lypemia. That one, those two you can really see. The lipemic, the chylomicrons, or the high lipid content, and the Icterus, the green. But the high protein, you can't tell. So what happened was I went over to chemistry and asked, well, what was the protein content? And it was, normal protein is like 7. I think the protein was about 8 or 9. That's high. That's a high serum protein level. Okay. And then spherocytes. I told you, you can get a high MCHC. It'll probably be about 37 or maybe 38 at the most. You can't correct for those. Okay, so you'll repeat it and it won't correct. Rule of threes will probably pass, but when you look at the smear, then you'll see that you're actually dealing with spherocytes. So those are instances where you will have high MCHCs. Remember, normally, with the exception of spherocytes, a high MCHC is not reportable, and you have to resort to troubleshooting. Okay, spherocytes, seen in hereditary spherocytosis. It's seen in hemolytic anemia and post-transfusion. Okay, so in hemolytic anemia, hereditary spherocytosis, that's the disease, and the type of anemia is hemolytic anemia, because there's different types of hemolytic anemia. So in spherocytosis, you have a lot of spherocytes. So remember the shape of these cells. These are spheres. Okay, it's not biconcave disc. These are balls. These are spheres. So the maximum volume has already been reached. So if the maximum volume has already been reached, what do you think will happen to them if you stress it out? Now, they're not going to be so pliable and flexible that can go through the sinusoids in the liver, because these are spheres. But what do you think will happen when it tries to go into these sinusoids? You think it'll have a hard time? Anybody? It's one or two answers, yes or no?
Yeah.
Yes or yes?
Yes.
Okay. So what happens is these cells are susceptible to stress. Okay. So what happens is when they go through the liver or whatever, or the spleen, and they're forced to go through those sinusoids, they actually burst. Okay, so what you have are called schistocytes. I'm going to get into schistocytes later. Schistocytes are fragments of red blood cells. Okay, fragments of red blood cells. They don't look like the typical biconcave disc with a central pallor, and obviously they don't look like the spheres because they've already burst. Okay, so what happens is you'll see schistocytes. You'll see that term later on. That's what happens in hemolytic anemia. Hemolytic anemia, there's two ways that can cause you to have anemia. Okay, two ways that can cause you to have anemia. One is decreased production. You already saw that with the iron deficiency anemia. That's decreased production. Another way to be anemic is increased destruction. If you have increased destruction of your red cells, that will cause you to be anemic. In this case, if it's destruction, it's hemolytic anemia. Okay. There's different types of anemia, and I'll explain to you what's going on in each of the different types. Okay, spherocytes. You see these small dark cells here, here. You notice that they're smaller, like I pointed out, and two, if you look at, they're darker and no central pallor. No central pallor. These are spherocytes. They will with this amount present, it's probably hereditary spherocytosis. Okay, so these are spherocytes. They're small, they're a little bit dark, not blue, but they're dark, and there's no central pallor. This one you can't compare. This one I think is probably a macrocyte, yeah, it is a macrocyte. This one too, I look closely, it's almost like basophilic stippling, but I don't see too many cells here that are normal. But these are spherocytes. They're small, microcytic, no central pallor. Okay. Okay, that's it for spherocytes. The next one is echinocytes. Echino, the prefix means prickly or spiny. Echinoderm, which is the term for starfish. Okay, so echino means prickly or spiny, and these cells are also called crenated cells or Burr cells. And these projections, the spicules or the spikes or the spines, are evenly distributed all over the red cell evenly. Okay, they're evenly distributed. Crenated cells are if the spines are sharp. Okay, they're really sharp. Burr cells are the same thing, except the spines are soft. They're roundish. So they're really sharp, or they're really soft and rounded. Okay, so in my training, I had to distinguish between crenated and burr. The crenated are the sharp spicules on the cell, and the Burr cells are like bumps, evenly distributed on the surface of the red blood cell. Crenated cells is an artifact due to faulty drying. Conditions that produce echinocytes is uremia. That's urea in the blood. You don't want too much. That's urine in the blood. Heart disease, dehydration, untreated hypothyroidism. So it's your water imbalance. Usually it's caused by water imbalance like dehydration. Okay, if you're dehydrated, then chances are you could probably have crenated or Burr cells in your blood. And this is what they look like. See these spikes? I kind of don't like this slide, but these are, see how the spikes are on, all over the surface on the blood of the red blood cells. See, these are sharp, except for over here. See how the spikes are soft? This would be called a burr cell. Okay, and this one is, they're sharp, so these are called crenated cells. Burr cells and crenated cells. Okay, so remember those two terms, crenated and burr cells. Crenated, they're sharp, and burr cells, they're soft and rounded, just like this one. Okay. Acanthocytes. Okay, acanthocytes are almost look like the crenated cells. These are red blood cells with 3 to 12 irregularly spaced spicules. Okay, the spicules, I mean, in the crenated red blood cells, the spicules or the spikes are evenly distributed across the surface of the red blood cell. For acanthocytes, they can be 3 to 12, but usually when I see an acanthocyte, it's usually about 1 or 2 or 3. Okay, and what they look like are like, they look like finger-like fingers sticking out of the red blood cell, finger-like projections. Okay, and these are acanthocytes. The spicules vary in length and are blunt rather than pointed. So they're not blunt like the burr cells, but they look like fingers, if you can remember that. The cause of acanthocytes is one of two conditions. The first one is, and I'm not going to make you memorize this term, but it's lecithin cholesterol acetyltransferase, or LCAT. You can say LCAT. So what can cause acanthocytes, or what type of cell would you see in the presence of an LCAT enzyme deficiency? And then you would say acanthocytes. And what this means, it leads to excess cholesterol in your blood. Hopefully in chemistry, you'll talk about the lipoproteinemias, and you'll talk about LCAT, or lecithin cholesterol acetyltransferase. So what happens is the excess cholesterol will lead to acanthocyte formation, or it could also lead to target cell formation, and I'll get to target cells later. The other condition that causes acanthocytes, and this was the one that was asked on my board exam. Okay, so A beta lipoproteinemia will result in what abnormal red cell in the blood, and it was A beta lipoproteinemia. That one we had to know how to spell. So that means A beta lipoproteinemia. If I had a board, the A means it's missing. Beta means it's a beta type of the protein. Have you guys gone over electrophoresis yet in chemistry? Does that sound familiar? When you fractionate a protein, when you fractionate a protein due to electrophoresis, that means it's taking a protein molecule and subjecting it to an electric current. So what will happen is, on electrophoresis, is that the current will separate the different fractions in a protein molecule. And the different fractions are albumin, alpha 1, alpha 2, beta, and gamma. Okay. Gosh, I wish I had a board. But what happens is, in the beta part, this is the beta lipoprotein, is in the beta portion of the protein molecule. And then the lipoprotein is a lipid protein. And anything that's anemia, that means it's in the blood. So it's a missing beta protein in the blood. Missing beta lipoprotein in the blood. So that's a breakdown of that word. So whenever you have A beta lipoproteinemia, that's also known as acanthocytosis. You'll see acanthocytes in the blood. And this is a condition where the fat and the fat-soluble vitamins cannot be absorbed from food. So the fat-soluble vitamins cannot be absorbed. And those vitamins are A, D, E, and K. So the fat-soluble vitamins cannot be absorbed. So ADEC is acronym, and those are your fat-soluble vitamins. Make sure you know this, and it's in red. You'll be asked it for sure. Okay, what are the fat-soluble vitamins? A, D, vitamin E and vitamin K. Okay, so here's the acanthocytes. See, it's almost like the echinocyte or the crenated red blood cell. So it's unevenly distributed. They look like fingers sticking out of the red blood cell. Okay, these are the unevenly spaced spicules. These are acanthocytes. This is, see how, like this one here, this is a pretty good looking burr cell. See how it's evenly distributed, but it's not sharp. These are not sharp spicules. They're like rounded and blunted projections sticking out of the red blood cell. Okay, this is called a burr cell. This one here and this one here are called acanthocytes. Okay, unevenly distributed across the red blood surface of the red blood cell. So these are acanthocytes. They look like fingers. Okay, remember that. A beta lipoproteinemia and LCAT. Remember that. The next one is teardrop cell. Just like the term, teardrops look like tears. Okay. So I'm going to be asking you the alternate names of these cells. The acanthocyte and the spherocyte don't have alternate names, but the teardrop cell has an alternate name. It's also known as dacryocyte. It's either dacryo or dacrocyte. I think when I learned it, it was the Y was there. So if you can see the cry in the middle of this term, that'll help you learn that a dacryocyte is a teardrop cell. It's pear-shaped. It's a cell that's sharp on one end. It's pear-shaped. Okay. Dacryocytes are associated with thalassemia and iron deficiency anemia. Anything is associated with iron deficiency anemia because in that type of anemia, you'll see all kinds of poik. Okay, you'll see teardrops, you'll see target cells, you'll see schistocytes, you'll see microcytes, macrocytes, you'll see the whole spectrum in iron. That's why I like iron deficiency anemia, because you see the whole spectrum. Thalassemia, you'll see mostly targets, but you can also see target cells in there as well. Okay, so dacryocyte, cry, teardrop cell. Make sure you know the alternate name. Like I said, it's pointed on one end. Here, these are, it's obviously, they look like tears, okay, pointed on one end. These are dacryocytes, or teardrop cells. Okay, bite cell. A bite cell is an interesting cell because this type of cell, the spleen, has taken a bite out of the red blood cell. So one of the RBC inclusions, an RBC inclusion is something that's seen inside of the red blood cell. So one of the RBC inclusions are called Heinz bodies. Heinz bodies are formed when hemoglobin is damaged by oxidants. Okay, oxidants are things that are toxic to your body, chemicals or toxins that harm your body, specifically hemoglobin. So when in the presence of oxidants, excuse me, an electron from the hemoglobin transfers to an oxygen molecule. Okay, you know, hemoglobin and oxygen, the relationship, right? Well, an electron from hemoglobin is transferred to an oxygen molecule. And what that forms is a reactive oxygen species. Okay. So you have this damaged hemoglobin that's been damaged by oxidants. An electron from this damaged hemoglobin will transfer to an oxygen molecule that is supposed to be carrying, right? Well, what happens to that oxygen molecule, it becomes what's called a reactive oxygen species. Okay, that's in the hemoglobin. And it's the reactive oxygen species, the ROS, that causes cell damage and lysis. So the ROS is toxic, it's harmful to the individual. Okay. These damaged cells that contain the Heinz bodies, the reactive oxygen species. So you know how you get the reactive oxygen species, right? Hemoglobin is damaged and electrons transfer to oxygen, forming a reactive oxygen species. Okay, these are damaged cells, cells that contain Heinz bodies. Your body is smart, and I'll keep saying that throughout the whole course, is that your body is smart and it wants to get rid of these damaged cells. These cells with the Heinz bodies are removed by macrophages in the spleen. Remember, macrophages are part of the reticuloendothelial system. They're like the garbage trucks. They collect junk and bad things in your body. They just clean up your body, take it out, and make sure it's not in circulation. So what will happen is the macrophages in the spleen will take out the damaged cells in the spleen. So what happens, that the spleen will actually take a bite out of the red cells, the Heinz body portion of that red blood cells, and then you end up with what's called the bite cell, okay? Bite cells or Heinz bodies is associated with a condition called G6pd deficiency, glucose-6-phosphate dehydrogenase deficiency. So this enzyme, G6pd, glucose-6-phosphate dehydrogenase, is very important because you need it to process sugars in your body. If you don't have it, then you won't be able to properly process sugars in your body, okay? So your body needs G6pd. But in patients who don't have the enzyme, the condition is called G6pd deficiency. Then you're prone to having your hemoglobin being damaged by oxidants, and you're creating Heinz bodies in the ROS, etc. Okay, you need to have the G6pd to prevent from having Heinz bodies. Okay, so G6pd deficiency primarily affects males and is the most common in people of African, Asian, or Mediterranean descent. G6pd deficiency. Individuals with this deficiency, the G6pd deficiency, can experience harmful health effects if they're exposed to certain foods, chemicals, or drugs. Those are the oxidant drugs, okay? If you're exposed to these chemicals, certain chemicals or drugs, like for example, primaquine, or what's the one for COVID, hydroxychloroquine, sulfa, can cause illnesses. These are oxidant drugs. These drugs will make the person sick, okay, because they don't because of the G6pd deficiency. So in the military, all people entering the military are screened for they used to be screened for G6pd because of the potential of them being deployed into areas that have a high concentration of malaria. So if a certain location has a high concentration of malaria, then they're given prophylactically the hydroxychloroquine. If you have G6pd deficiency, then you can't take hydroxychloroquine or the primaquine. Okay, so it's if you're deficient, then it's automatically a disqualifier for joining the military. So you need to have G6pd. If you have a deficiency of this enzyme, then you're prone to creating the Heinz bodies and the ROS and creating bite cells. So individuals with G6pd deficiency, when exposed to oxidant drugs, will form Heinz bodies. And the Heinz bodies, when the red cells, remember what happens, the red cells that has the Heinz bodies, when it goes through the spleen, what's the spleen going to do? It's going to take a bite out of the red cell. Therefore, you have bite cells. Okay, so what you need to do is to associate Heinz bodies with bite cells with G6pd deficiency. Make sure you associate those three together: Heinz bodies, bite cells, and G6pd deficiency. All right. And there's a bite cell. And so a spleen's taking a bite out of this red blood cell, hoping that it will take out the Heinz bodies. There's another cell. There's another cell that looks like this cell, and it's called a helmet cell. I'll get into that very shortly, because the next section are schistocytes. I talked about schistocytes and hemolytic anemia. When spherocytes are destroyed, or actually any kind of red blood cell, it doesn't have to be a spherocyte, but you can have normal red blood cells that can be destroyed by mechanical destruction. Say, for example, if you had a new heart valve put in, your body's not going to be used to it, or the valve is not used to it. So you're going to be pumping your blood through this new heart valve, and it's going to pound on the red blood cells, destroying the red blood cells and creating fragments or schistocytes. Okay. Another way to form schistocytes or destruction of red blood cells. Remember, there's one of two ways to have anemia: in decreased production of red blood cells, that's anemia, or increased destruction. Remember those two statements: decreased production or increased destruction. So schistocytes are formed by increased destruction of red blood cells. Schistocytes are associated with DIC. I'm going to get into that extensively in the coag section of this course. Hemolytic anemia, I already talked about hemolytic anemia, but there is a lecture dedicated to hemolytic anemia. Heart valve surgery, I just mentioned, like if you have a new heart valve, that valve can actually destroy red blood cells, causing schistocytes. And severe burns. If you're in a burn, the burn can actually create schistocytes. The difference between schistocytes are small. These are small cells. They really don't have any function, but they're just really fragments of red blood cell. These are small and they have no central pallor, and that's an important thing to note. Another thing that's important to note is that schistocytes can take on many shapes. Okay, so you have keratocytes, and these are cells that have horn-like projections that are called keratocytes, or triangulocytes. They take on the shape of triangles, or they take on the shapes of a helmet, and they're called helmet cells. So whatever they take on, whether it looks like a horn or a triangle or a helmet, these are still all schistocytes. I don't know, whatever shape you can be created. That shape is created by randomly because of the how the cell was destroyed. It can be keratocytes, triangulocytes, helmet cells. Those are just three of probably many types of shapes that can be created as the normal red blood cells is destroyed. Now, helmet cells are in particular. There's confusion between the helmet cell and a bite cell. Okay, because helmet cells, they look like a helmet, they also look like bite cells, but helmet cells are schistocytes. These are cells that just happen to look like helmets, okay? And the way that you can tell the difference between a helmet and a bite cell is that a bite cell has central pallor and a helmet cell does not have central pallor. And remember, helmet cells are smaller. Bite cells are normal size with a bite on it, and it should have regular central pallor. But helmet cells, like I said, is a destroyed red blood cell and does not have central pallor. So that's the difference between a helmet cell and a bite cell. Okay. Okay, this is a helmet cell. It's supposed to be smaller, but you can see how that can look like a bite cell, but it's actually a helmet cell. Schistocytes. Okay, schistocytes, these fragments here, small fragment, I guess that can be a fragment, that could be a fragment there, but this one and this one are definitely schistocytes. It's reportable. Okay, so if you see schistocytes, if you see POIC, then you'll say, okay, 1 plus 2 plus of schistocytes are noted. Okay. Target cell. Target cell has an alternate name and it's called codocyte. Okay, target cell. And it looks like a target. It looks like a bullseye target. Okay, it's centrally stained area where the central pallor is supposed to be. These cells are usually hypochromic, meaning has a low hemoglobin content. So the shape the shapes of the red blood cells. So if I ask you, what is the shape of a normal red blood cell? What would you tell me?
Biconcave.
Correct. Biconcave disc. I could ask you, what is the shape of a target cell? You would tell me that it's bell-shaped. Okay. Now you wouldn't say target cell. Well, it looks like a target. No, I'm asking for you what the shape of the actual cell is. The shape of a target cell is a bell shape, but on the smear, when it's stained, it looks like a bullseye target. Okay, because the targeting occurs while it's drying. It's almost kind of like an artifact. That's what it looks like. Target cells, the T for target, just remember that T for target is seen in thalassemia. Thalassemia, the predominant cell type is our target cells, but it can also be seen in liver disease, sickle cell disease. Predominant cell type in sickle disease, obviously, are sickles, and hemoglobin CESC disease. I'll talk about these three diseases and sickle cells in the hemoglobinopathy lecture. You'll learn more about those types of hemoglobins. But for now, target cells, thalassemia, okay? If you're asked the question, what disease or patient has thalassemia, what would be the predominant cell type seen on the peripheral smear? Target cells are microcytic. Just remember that. They have a low MCV. Thalassemia, the type of, I don't know if you remember, the type of anemia, again, when I say type, it could be either megaloblastic, microcytic, or hypochromic microcytic, okay? So thalassemia is a hypochromic microcytic anemia, and predominant cell type are target cells. Just like that. Just like bullseye target, bullseye, bullseye. These are target cells. Alternate name, anybody remember?
Codocytes?
Codocyte, okay?
Oh.
Codocyte. What's the alternate name of a teardrop cell?
Dacrocyte.
Acanthocyte. Good, thank you. All right. Okay, sickle cells. Alternate name for sickle cell is drepanocyte. Unfortunately, I don't have any trick for drepanocyte and sickle cell. You'll just have to know it. So drepanocyte and sickle cell. So the shape is sickle or crescent shape. These are pointed on both ends. They're really skinny, pointed at least one end. Actually, it's pointed on both ends. If it's pointed on one end, then that's a dacryocyte, okay? Drepanocyte or sickle cell is associated with hemoglobin S, and then it's seen in sickle cell anemia. So drepanocyte is the alternate name. Make sure you know that. Just like that. You can see how patients who have sickle cells have difficulty breathing, not too much space for hemoglobin and oxygen content. If a patient has a lot of these sickle cells on the peripheral smear, and that's called sickle cell disease. Okay, that means the patient is in crisis. Okay, so that's sickle cell disease. If a patient only has a few or occasional sickles like this, the patient has sickle cell disease, but it's said to that patient has sickle cell trait. So there's a difference between trait and disease. The trait will have few or occasional numbers of sickles in the blood, but if you have full-blown sickle cell disease, then you have a significant number of sickle cells in the blood. Okay, so there's a difference between trait and disease. Have you guys heard of the term homozygous and heterozygous? What does homozygous mean?
Same.
Same. Okay. So in terms of genetics,
They have the same trait.
have the same trait. Well, they have the same, actually they have the same gene. Say for example, okay, let's take blood types. If you're if your blood type is AO versus AA, what is your blood type? If you're AO, what is your blood type? You're type A, okay, because O is called recessive. And if you're AA, then you're also type A. The AO is called heterozygous because you have the A, and then you also have the O gene, but the O is recessive. It's not really expressed. So the phenotype, which is what you really see, is the A because it's the dominant. AO, what will be seen is type A. If you're AA, then what you'll see, the phenotype is type A. The AO is called heterozygous and AA is called homozygous. That means the alleles of the both genes are the same. In the case of heterozygous, they can be different, and what will show up is a dominant gene. So if you're heterozygous for the disease, usually if you're heterozygous, you'll show the trait, and if you're homozygous, then you'll show the disease, okay? That's the difference between heterozygous and homozygous. You'll get a little bit more of that in when you take the blood bank course next summer. Okay, so I'm just giving you a little preview of homozygous, heterozygous, and trait and disease. So trait is mild form of the disease, and the disease is actually full-blown disease, okay? All right, the next cell is ovalocyte and elliptocyte. Again, this is something where in my training I had to know the difference, but basically the difference is really easy. Ovalocytes are egg-shaped, but they're stretched out, and the elliptocytes are cigar-shaped or pencil-shaped. They're really skinny and they're stretched out red blood cells. Okay, so these shapes are due to red cell membrane defect. And then you can have a hereditary ovalocytosis and hereditary elliptocytosis. So like I said, ovalocytes are more egg-shaped and elliptocytes are more pencil-shaped. Okay, so here, these egg-shaped red blood cells, obviously these are not normal red blood cells, and then here are elliptocytes. Ovalocytes here and elliptocytes here. If you have a normal smear and everything looks round, and then occasionally you'll see these large oval, that's another good indication. It's reportable that you're seeing oval macrocytes. So you can see that these cells are macrocytic, they're big, but in the presence of normal red blood cells, and obviously there are not too many normal red blood cells here, but if you see these oval shapes like this, and they're large, oval macrocytes are reportable, and oval macrocytes are what's actually seen in iron deficiency anemia, or I'm sorry, not iron deficiency, the B12 folate or pernicious anemia. Okay, forget I said iron deficiency. I apologize, I didn't mean to say that. It's B12 folate or the megaloblastic anemia. So oval macrocytes are seen in the megaloblastic anemias, and those are the pernicious anemia, vitamin B12 folic acid deficiency. So oval macrocytes or ovalocytes and elliptocytes. Some more elliptocytes, ovalocytes. Okay, it's a little bit better here. Nice central pallor in all of them, looks like. But still, I don't see too many. So this is probably hereditary of something, because I don't really see too many normal red blood cells here. Nice central pallor. All right, the next one, stomatocyte, stoma. Stoma, the prefix means mouth, okay? Stomatocyte, stoma means mouth. It looks like a mouth, it looks like it's smiling, okay? So, and that's the shape of the central pallor. So oval or rectangular central pallor. It looks like a mouth. It's mostly artifactual, okay? Stomatocytes are associated with alcoholism, and it's seen in a condition hereditary stomatocytosis. Just like this. See, it has a slit in the middle of it. It looks like a mouth. That's the central pallor. These are not hyperchromic. Remember, in hyperchromic, this would be wide open. Looks like a donut. Okay, but here when they look like slits like this, like mouths, that's called a stomatocyte. Now, when you do your abnormal red blood cells, unfortunately, I'm not going to be in the lab. That's Xavier's going to be doing that this semester. You're going to be looking for stomatocytes, and it's in the red blood cell box. So you'll have to start looking for those. What you'll do is you'll find the cell, you'll identify it, you call Xavier. I said, this is a stomatocyte, and then he'll sign you off. So what you'll have to do is you're going to have to look for these cells. Okay, the next one is Rouleaux Formation. Rouleaux Formation, or Rouleaux, just regular Rouleaux. High protein condition, multiple myeloma, and Waldenstrom's macroglobular anemia. And another one is chronic inflammatory disorders. So those two main ones, multiple myeloma and Waldenstrom's, are conditions where you have a high protein content. High protein content. So if you spin down the blood, you have a high MCHC, Rouleaux fails, and you spin down your blood and it's clear, you'll have to suspect that it's probably due to a high protein content. Then you'll do a saline replacement. Okay, and then your MCHC will come down and your Rouleaux will calm down and your Rouleaux will pass. So this is what happens in Rouleaux formation. The term is stack of coins. Stack of coins. Okay, it looks like a stack of coins. So they're one behind the other. Okay, like a stack of coins. And this is Rouleaux formation. They stick. If you want to see Rouleaux formation, you can see Rouleaux formation if you go too deep. Okay, so when you're looking at your smears under the microscope and you're with Xavier in the laboratory, you need to be looking towards the end of the feather end, almost to the tip, not where the cells are too sparse, but when they're almost touching. It's almost like this. You need to look at a field where the distribution of red cells is kind of like this. If you go too far deep into the feathered edge, you're going to actually see Rouleaux formation, but it's not Rouleaux. Okay, so if you want to see what Rouleaux looks like, go deep into your smear and take a look at the red cells. The cells will actually stack up like this, but it's not Rouleaux formation. That's why you need to be near the edge, near the end of the feather edge of your smear. You don't want to be reporting that false Rouleaux when you go too far deep in. Okay, so that's what Rouleaux looks like, as opposed to agglutination. Okay, so red cells will stick together in the presence of high protein, and that's what you get. You get Rouleaux. But in the presence of antibodies or cold agglutinins, you get agglutination. So here, red cells will also stick. Okay, so here, Rouleaux formation, high protein content. How do you correct for this? You correct it by doing a saline replacement. In agglutination, due to cold agglutinins, due to the anti-I antibody, the red blood cells will be sticking to each other. It's due to the activity of this anti-I antibody. This antibody is a cold antibody. When you get into blood bank, you'll know that there are two types of antibodies, cold and warm antibodies. Anti-I is a cold antibody. That means that it's active in cold temperatures. So when you draw a patient's blood, okay, it's obviously at 37 degrees. But when you take it down to the laboratory, if you take the long way back to the laboratory, it's cooling down, okay? So that means it's cooling down. The anti-antibody could be acting up. So by the time it gets to the hematology department, the temperature is probably much less than 37 degrees. So when you run the specimen on the analyzer, you're going to be getting the clumping of, you're going to get red blood cells with an active anti-I antibody, which is going to cause the red blood cells to be clumped. And it looks like that. See, the red cells will be sticking together like this. See, it's not like Rouleaux, but it's just because of the activity of anti-I. Anti-I is on the surface of the red blood cells, and it's causing the red blood cells to be sticky to each other. Because anti-I is a cold antibody. The correction for this is to pre-warm the tube. Remember I said one of the easy ways to troubleshoot a high MCHC? This is a really common one, and it's a real easy one to fix. You just take the tube, stick it in the heat block for half an hour, and repeat the test. Because once you put it in the heat block, you can do a whole bunch of other things. And then after half an hour, then you'll run the tube, you'll mix it, and then you'll run the tube, and then everything, because anti-I is no longer active at 37 degrees. Therefore, the red cells will not stick, and then you'll get a nice, clean CBC. Normal CHC and your Rouleaux 3s will pass again. Another thing too, when you have this, and you run it through your analyzer, your MCHC will be high, your MCHC will fail. What else do you think will be elevated super sky high? What other CBC?
MCV?
Say again.
Hemoglobin.
What is it?
Hemoglobin.
Hematocrit? No, not really, because when you're gonna lyse the cells, and you're gonna get your red cells no matter what. Where there's packed red cells, or you're gonna get an accurate hematocrit. But when the red cells are sticking together and they're going through the flow cell, what do you think the instrument's gonna read?
High MCV.
Yeah, you're gonna get a high MCV. So here, you see, you're gonna get, what's the normal value for this red blood cell? It's like 70 to, or 80 to 100, right? You think this is 80 to 100 when it goes through the flow cell? No, you're gonna get a super sky high MCV, and that's what your instrument's gonna read. Even this, they're stuck together. When you run this through the analyzer, it's going to read it the same way it reads this red blood cell or this red blood cell. This normal for that one is 80 to 100. This is not 80 to 100. You're going to get an incredibly high MCV. Okay, so just keep that in mind. Just think about what's going on when you see what doesn't make sense when you look at your CBC. When you have it in your hand, if you see a MCV 150, 160, I said, no way. That's impossible. That's physically impossible. That's not like this. That's a humongous site. Okay. So you have to correct for that. So the correction for cold agglutinin is what?
Warming it.
Warming it. Okay. The correction for Rouleaux formation is doing what?
Saline replacement.
Saline replacement because of the high protein. What is the correction for Lypemia?
Saline replacement.
Saline replacement or Icterus. Icterus, person with liver problem.
Same thing. Saline replacement.
Same thing, saline replacement. So that's why it's good when you're on the bench and you have things that are suspicious. It's legal for you, and it's not HIPAA. You can call the flora and say, I have a problem with this CBC. Can you tell me what the patient's diagnosis is? I'm troubleshooting a specimen. And they should be able to tell you. I hope they haven't gotten stricter on that. But when I was on the bench and I had a problem with CBC and I wanted to find out what's going on because I have really screwy abnormal results, I would call the floor and ask, what's going on with this patient? Can you give me a diagnosis? And then whatever they tell me would make sense. Okay, I mentioned inclusion bodies. Inclusion bodies are things that are seen within the red blood cell. It's included within the red blood cell. That's why inclusion bodies. Howell-Jolly bodies, I talked about Howell-Jolly bodies in a previous lecture. Pappenheimer bodies I'm going to talk about. Basophilic stippling are inclusion bodies, and I'm going to be talking about that. Cabot's rings, I touched on Heinz bodies and parasites. Okay, so unfortunately, when you look at your peripheral smears on the bench in hematology, Not all your cells or not all your smears are going to be perfect. You know, it's not going to be that boring. You're going to be seeing things like this. Basophilic Stippling, Heinz, I don't know. These are actually kind of rare, but you're going to see different types of shapes. You'll definitely see different types of poikilocytosis. But you need to be prepared to identify Howell Jolly bodies, Pappenheimers, especially basophilic stippling, Cabot's rings, Heinz bodies, and parasites. Okay. Howell Jolly bodies. Howell Jolly bodies are seen in the right stain. The type of stain that you see in the normal hematology laboratory, the stain is called a right stain. I think you're going to be having a staining lab tomorrow. I'm not quite sure, but you're going to be doing the Diff-Quick staining procedure, where you're going to take your smears and you'll actually stain them. Hopefully, you'll be able to look at the cells underneath it. Howell Jolly bodies are made of denatured DNA or DNA remnants. Okay. And these are multiple bodies that in the plates. Usually they're single, but multiple bodies of Howell Jolly bodies. If you see more than one within a red blood cell, then that means you have a megaloblastic anemia. A megaloblastic anemia, remember, is a type of anemia. And the specific anemia is vitamin B12 folate deficiency or pernicious anemia. That's the actual anemia. Megaloblastic anemia is not the name of an anemia. It's a type of anemia, okay? Make sure you remember that. Howell Jolly bodies are seen in patients with splenectomy, megaloblastic anemia, thalassemia, and hemolytic anemia. The splenectomy is the more common one. A person who has their spleen removed, they have a tendency to have Howell Jolly bodies in their peripheral blood because the spleen's gone. Usually the spleen will take care of the denatured DNA or DNA remnants, and the spleen will go ahead and remove that. It doesn't remove it like it removes the Heinz bodies in G6pd deficiency, where it just takes a bite out of it and says, well, you know, I'm going to take a chunk out of it, and that's it. That's too bad for you. The splenectomy, the denatured DNA is removed a little bit more gently by the spleen, and without the spleen, then you'll see Howell Jolly bodies. Okay, so Howell Jolly bodies are denatured DNA that's normally taken out by the spleen, but without the spleen, then you'll be able to see them. And that's what they look like. Small, perfectly round black marbles. This is a nice, good one here. Okay, small and perfectly round. I think you'll see Howell Jolly bodies in the laboratory. The difference between, and it gets confused with staining granules or platelets. Platelets are not perfectly round, or staining precipitate. And the way you can tell the difference between, okay, so here's a platelet here. This platelet can be on top of this red blood cell here. And what will happen is, if you see around it a clearing, like a little halo around it, that means it's an artifact. Because you can't see, well, you can almost see it here, but it's not. But here, you don't see a clearing or a halo around this Howell Jolly body. Therefore, this is real. If you did see a clearing or a halo, that means whatever you're looking at is actually not inside the red blood cell. It's like it's on top of it. So it's an artifact. Okay. Usually platelets or staining granules are on top of the red blood cell, and it looks like Howell Jolly bodies, but it's not. But make sure that you see perfectly round dark black marbles. Okay. Those are the Howell Jolly bodies. Denatured DNA. Pappenheimer bodies. Pappenheimer bodies are made out of iron. Okay, they're made out of iron, and they're usually found in clusters that are small, and it's usually near the periphery of the smear of the cell. So Pappenheimer bodies are made out of iron. Pappenheimer bodies are seen in right stain. Okay, Pappenheimer bodies are seen in right stain. These Pappenheimer bodies, if you stain it with Prussian blue, a different stain as opposed to right stain, then what you're actually seeing are siderotic granules. Okay, siderotic granules if you stain it with Prussian blue. It's the same thing like reticulocytes. If you see polychromasia, Remember, if you see polychromasia, that's a term using right stain. However, if you use the new methylene blue, then what do you call it? Then you call it a reticulocyte. Okay, so using a different stain, the term is different. Using a right stain, the term is different. So right stain, the term is Pappenheimer bodies. Using the Prussian blue, it's called siderotic granules. Okay, just like remember on for the red blood cells, right stain is polychromasia, and new methylene blue is called reticulocyte. Okay, so make sure you know that. So under different staining conditions, you have different terms, just like the red blood cell and retics. So Pappenheimer bodies are associated with sideroblastic anemia. Remember, it's iron again, right? Sideroblastic anemia, sickle cell anemia, thalassemia, and following splenectomy. These are like iron deficient, and iron deficiency anemia is not in here, but it's an iron associated with iron metabolism. So sideroblastic anemia, you'll see aggregates of iron, okay, siderotic granules, and Pappenheimer bodies. And it looks like this. It's not really here and here. These are granules that are near the periphery of the red blood cell. These are Pappenheimer bodies consisting of iron. So if you use the Prussian blue stain, remember the Prussian blue stain, it's siderotic granules. Otherwise, it's known as Pappenheimer bodies on the right stain. Okay, basophilic stippling. Basophilic stippling is important to note. It's very reportable. That's what's seen on right stain. And basophilic stippling is precipitated ribosomes. So you got denatured DNA, you got iron, and then you have precipitated ribosomes. Make sure you know what the inclusions are made of, because you're going to be asked. So basophilic stippling is precipitated ribosomes. Precipitated ribosomes, and basophilic stippling is under right stain. And basophilic stippling occurs when the smear is drying, and it doesn't occur in vivo. Like if you're in the vein and you're looking for basophilic stippling, you're not going to see it. You'll only see it after it's taken. Its peripheral blood is taken out. You make a smear, and the smear is drying. So the basophilic stippling occurs in the drying process. The important thing to note about basophilic stippling, and doctors are really, they want to know if basophilic stippling is present, especially in pediatric patients, because a long time ago, like in the 60s or 70s, when families were living in the low income, low rent housing, those types of homes were painted with paint that contained lead. And as the paint was chipping, the children, I don't know, they would eat the chips. Okay, children eating paint chips, and as a result, they got lead poisoning. So they drew their blood, and they saw a lot of basophilic stippling. So basophilic stippling is associated with lead poisoning, not always from kids eating paint chips, but lead poisoning. Okay, there's other ways that you can get lead poisoning, but one of the first ways that it was discovered on basophilic stippling was children eating paint chips. So make sure you know that basophilic stippling is caused by lead poisoning. Another place where you'll see basophilic stippling is in leukemia. So that's why basophilic stippling, if you see it, it's important for a couple reasons. One, you have a possible lead poisoning situation, or two, if you say increased basophilic stippling, you could be saying that this patient might have leukemia. But with leukemia, you'll probably have to associate that with a high white count. Remember, you cannot make a full diagnosis on one lab test. Okay, so basophilic stippling can be in a couple conditions: lead poisoning and leukemia. Okay, and it's really reportable. You report it, you make sure you know what you're reporting. Okay, so here it's difficult to see. First glance, you'll say, I don't see anything. But if you look closely at this cell here and this cell here, they look like freckles, okay? It's evenly distributed around the cell, not like the Heinz bodies or the Pappenheimer bodies where it's off to one side. You have an aggregation where it's off to one side. Basophilic stippling is throughout the entire cell, throughout the entire cell. If you see that, that's called basophilic stippling. You'll be seeing this on a test. This slide, and you need to be able to identify it. Okay, so this is basophilic stippling. If you see a lot of it, actually, you know what? Two in one field is actually a lot. So this is fortunate that you're seeing basophilic stippling on this particular slide. But the granules, remember, basophilic stippling is precipitated ribosomes, and it's found throughout the cell. Could be lead poisoning, or it could be leukemia, but make sure leave that to the doctor to decide. So it's very important, if you see it, to report it. Cabot's rings. Cabot's rings are thread-like loops. You don't see too many. I haven't seen too many Cabot's rings. If I did, I can't remember. I think I may have seen it once. Thread-like loops or figure-8 filament. Okay, Cabot's rings are seen in right stains, and Cabot rings are composed of remnants of the microtubule. Microtubule is, you know, when you have mitosis, when you have cell division, you know, and the nucleus splits apart. Well, you know, those strands between the nucleus, it's called the microtubule. So those are remnants of the microtubules that just lingers around. Okay, it's indicative of abnormal erythropoiesis. So Cabot rings are seen on right stain. It's associated with megaloblastic anemia, postsplenectomy, homozygous thalassemia. Okay, that's what it's seen in. If you notice, not too much red on this slide. Cabot's rings is difficult to see. This is a Cabot's ring. I think there's another slide with Cabot's ring, and it's like right here. This almost looks like, to me, it looks like basophilic stippling, but it's not. Okay, and that's it for Cabot's ring. This, what do you think this is? Anybody? Anybody care to venture to guess?
Polychromation.
NRBC.
Say it again,
The nucleus?
louder.
NRBC.
Yes, that's an NRBC. The
Yeah.
thing about nucleated red blood cell, it never fails. The nucleus is perfectly round and it's jet black. Okay, I almost want to call this one here a not Dolly body, but a Howell Jolly body. But it's perfectly round and it's black, and it has the red blood cell around it. But this is a nucleated red blood cell. Okay, all right, good job, Ray. And this is Cabot's ring. I may or may not ask that, but because it's difficult and it's pretty rare. All right, Heinz bodies. Heinz bodies are made of denatured precipitated hemoglobin. Okay, so you got denatured DNA. You have a whole lot of things to know what things are remnants of hemoglobin. That's what this is, denatured precipitated hemoglobin. Heinz bodies are seen in right stain. Oh, I'm sorry, not seen in right stain. In order to see Heinz bodies, you're not going to see this on the bench. You have to use crystal violet or brilliant crystal blue. So of all the inclusion bodies, Heinz bodies will not be seen in right stain. You'll see the Pappenheimers, but you won't see siderotic granules. But Heinz bodies, you need to use a right stain. So if you have a person who has like G6pd deficiency, you might want to stain their blood using crystal violet or brilliant crystal blue and start looking for Heinz bodies. Okay. Remember, Heinz bodies are taken out by the spleen. called a pitting mechanism. So it takes a bite out of the red blood cell, leaving a bite mark, if you will. It's associated with G6pd deficiency and alpha thalassemia, mainly G6pd deficiency. So remember, Heinz bodies, bite cells, and G6pd deficiency are all associated with each other. Bite cells, Heinz bodies, and G6pd. So this is one of those stains. It's either crystal violet or brilliant crystal blue. And you can see it's not a right stain. And this, to me, I don't know if you've done the retic lab yet, but these retics look like this under the new methylene blue. But these are what Heinz bodies look like. So this person has G6pd deficiency. Okay, so that's it for those inclusion bodies. The next one is malaria. Malaria is the most common protozoan infection. It's the most common protozoan infection. Protozoan means those are the small guys, and they're small parasites. And there are 4 species of Plasmodium: Plasmodium malaria, Falciparum, ovale, and vivax. So when you take microbiology next semester, I'm going to show you how you can speciate, how you can tell the difference between a Plasmodium vivax infection versus a Plasmodium ovale. I'll show you how to differentiate between the 4 species of malaria. And also too, you guys are going to be memorizing the malaria life cycle. What happens when a mosquito bites you, and what happens inside your body, inside the red blood cell. So you will be memorizing that, and that'll actually be kind of fun because you'll be competing against each other on and racing on putting that on the board. But that's next semester in micro. So you get this through a mosquito bite, obviously. When you see malaria, or when you suspect malaria, somebody from a malaria endemic country like in Africa or Southeast Asia or wherever, usually Southeast Asia, Malaysia, or especially Africa, you're going to be saying, okay, this is a malarial smear. When you get into micro, you'll see that there's 2 types of smears: the thick smear and the thin smear. When you're looking for malaria, you're going to be looking so hard that you'll be mistaking malaria for platelets, and it's easy to do. They're that small. Okay. So this is malaria. It doesn't look like much. It looks like a platelet on top of this cell, but this one, there's a ring form. What you're actually looking for are the ring forms. Here is a ring form here. Okay, if you see this, a ring with a nucleus, a ring kind of like a nucleus. This is actually called a signet ring. Signet ring. Type that way. Signet ring. Okay, that's the type of appearance it looks like. So there's a ring here and a ring here. This one here, the next slide. Oh my goodness. I thought I had more slides of malaria. Okay, so you get a ring form here. In micro, you'll see more malaria slides. But there's a ring here and a ring here. I apologize. I thought there was a better malaria slide. Okay, so the next parasite is Babesia. Babesia microti. And malaria is transmitted through a mosquito. Babesia is transmitted by a tick bite. Okay, Babesia microti. And this is what it looks like on a peripheral smear. This X formation, 2 nuclei, more than 1 nuclei here. But this particular one here is a classic description of Babesia. Okay, I think that's it. Are there any questions on these RBC inclusions? The white blood cells, there are inclusions on white blood cells, a lot of them, like Dolly bodies, our rods, but we'll get into that when we do WBCs. But those are the main RBC inclusions, and those are the main shapes. So make sure you memorize and know the alternate names. Make sure you know what the different inclusions are made of. Like Cabot's ring is remnants of microtubule. Okay, precipitated ribosomes. Consists of iron. Jolly bodies are denatured DNA. Okay, know the difference between agglutination, what causes the agglutination, which is the anti-I antibody, which is a cold antibody, or the cold agglutinins. This one's caused by protein, the Waldenström's or the multiple myeloma, stomatocytes, okay? Ovalocyte, elliptocyte, sickle cell, alternate name, drepanocyte, target cells, a codocyte, schistocyte, no alternate name, but know the difference between a helmet cell and a bite cell. This is a helmet cell, no central power. Schistocytes can take on different shapes. They're fragmented, so this is destroyed red blood cells caused by destruction. Bite cells, notice the central power here. So you'll be given this slide, and I think I'll ask you, what is this, a helmet cell or a bite cell? Or you'll just tell me what it is. G6pd, a couple times talked about G6pd and Heinz bodies and bite cells. Okay, you need G6pd to live, to produce sugars. In teardrop cell, dacryocyte, acanthocyte, two conditions, A beta lipoproteinemia and LCAT. Okay, acanthocytes, they look like fingers, okay? They look like finger-like projections, as opposed to echinocytes. They're all over the surface of the red blood cell. Echinocytes are formed when there's a water conditions like dehydration or excess, or it could be like excess water in the blood. Either way, you'll get some echinocytes. So you got water imbalance. Spherocytes, they're microcytes. Remember, spherocytes, low MCV, no central power, and if you see spherocytes, you also might want to look for schistocytes. Okay, I'm surprised there are not any schistocytes on this field. Usually, and that's what I was taught. If you see spherocytes, you also might want to look for schistocytes, because these cells are balls on the verge of bursting. And some troubleshooting here. So remember, first thing to look at is MCHC, and look at your H and H. Those are the two things you look at when you're handed a CBC, MCHC and H and H. All right. Any questions?
I have a couple questions.
Oh my God. Okay.
So, did we go over how reactive oxygen species is formed?
Yeah.
What's that?
So in the presence of oxidant drugs, hemoglobin is bound to oxygen, right? Hemoglobin is bound to oxygen. So in the presence of oxidant drugs, an electron from the hemoglobin is transferred to an oxygen molecule. And that electron is kicked out of the hemoglobin because of the oxidant drugs, because of the chemical, the toxin, okay? So your normal relationship is hemoglobin is going to bind to oxygen, right? But in the presence of oxidants like this, it's going to kick out an electron from the hemoglobin molecule to the oxygen molecule, and that's where you form the reactive oxygen species.
Oh, okay.
Okay, I mean, that's a good question. That's But now you understand why oxidants are bad. And that's why you take antioxidants. Antioxidants are good, okay? Because, I mean, this probably happens a lot. That's probably why you're, I don't know, there are theories why of aging. Oxidants causes aging and your skin to be, old and wrinkled or whatever. But what happens is the oxidants will kick out an electron from hemoglobin. This is the normal relationship, hemoglobin and oxygen. But in the presence of an oxidant, it's going to cause the hemoglobin to kick out an electron and transfer to an oxygen molecule, okay? And then that's why you have a reactive oxygen species. And that's bad. Reactive oxygen species are not good. And like I said, your body's smart. It's going to identify those and filter those out. And in the process of filtering them out, that's how you create the bite cell.
Okay.
Okay.
And so are bite cells and Heinz bodies the same?
A bite cell is a red cell without the Heinz body. The Heinz body part of the red blood cell was chomped off by the spleen.
Okay.
Okay, so Heinz bodies are in a red blood cell. When it goes through the spleen, the Heinz bodies are taken out by the spleen, leaving behind a bite cell. Okay.
Okay.
Any other questions? You said that you had a whole bunch of questions.
Oh, no, those were it. Thank you.
Oh, okay.
I have one.
Okay.
You made a statement earlier in the lecture about it. You said an increase in death of a specific cell and also a marked decrease of a value, and I didn't catch the statement.
Okay. It's anemia in general. Okay. Anemia is a decreased production of red blood cells. Anemia can be caused by two conditions: a decreased production, like iron deficiency anemia, for example, or megaloblastic anemia, or B12 folate deficiency, where you're not producing red blood cells. Okay, so anemia can be caused by a decreased production of red blood cells, or it can be caused by an increase in destruction. Either way, you have a net of not too many a net negative amount of red blood cells. Okay, you either don't produce them or they're destroyed. Okay, that's what anemia is.
Thank you.
Is that what you needed? Okay, anybody else? I'm going to be saying that statement later. I think you'll see it and it'll become, you'll be familiar with it. I think it's the hemolytic anemia lecture or the aplastic anemia lecture, where I talk about anemia can be caused by one of two conditions: either decreased production or increased destruction. So in decreased production, you don't produce red blood cells, or increased destruction. And there's lots of ways to destroy red blood cells. Any other questions? Last call. Tomorrow's quiz, I think you'll do pretty well. I mean, from the way I did your homework, and you all should have gotten your homework back already. And if you didn't get it, let me know. But from looking at the homework, just as a reminder, if you can just not use the Google Docs. Every time I try to get into Google Docs, either it'll ask for access. I mean, I just like to keep it straightforward and keep it in Word. So just try to stay in Word, not go to Google Docs. That just makes it easy. Most of you did it in Word, and they were all beautiful. They were all really good, and I was really happy with it. Okay, any last minute questions? So I think I'm getting the hang of recording this lecture, and I'll send you the lecture hopefully later today. All right. All right, if there are no more questions, you can look on your quiz tomorrow, and I'll see you next Wednesday. All right.
All right. Have a good night.
All right. Thanks. Take care.